After identifying candidate CREs that are responsible for some of the observed differences between surface fish and cavefish, the authors wanted to delve into one of the cis-regulatory elements, CRE15, which is analyzed throughout Figure 5. Shown here is a map of chromatin accessibility across the gene HPDB. This gene is responsible for catalyzing the first step of tyrosine catabolism and is upregulated in cavefish. The gene HPDB contains CRE15, which is highlighted in the red box. As you can see, there's significant differential chromatin accessibility between cavefish and surface fish in this region. Shown below is a sequence containing the deletion that codes for the loss of the predicted binding site, which may be responsible for the observed differential chromatin accessibility. The next step was to find out what CRE15 is responsible for. Authors wanted to compare HPDB expression across three genotypes: the surface fish homozygote, the cavefish homozygote, and an F1 hybrid cross. As this graph demonstrates, the hybrids display an intermediate amount of expression, demonstrating the fact that these alleles share an incomplete dominance relationship. Here, the authors are determining whether the mutation is in cis or trans. Let's start with an overview of the two. Cis regulatory effects regulate expression of genes on the same allele. Trans regulatory factors modify the expression of distant genes. These chromatograms show sequencing in the HPDB gene. Because of the established incomplete dominance relationship between the surface and cavefish alleles, one should expect to observe transacting effects in the expression of another hybrid allele in the protein coding mRNA. Any differences in the expression between the DNA and mRNA are the result of a cis-mediated repression, since the mutation affects expression within the same gene, meaning this here is a result of a mutation in cis. After the authors detected the deletions in CRE15, the next step was to figure out what was being altered because of the deletion. Their initial prediction was that the deletion took place in the IRF2 repressor region. In order to test this, they ran a gel assay where proteins are tasked with binding to specific sequences. The authors isolated IRF2 binding sequences in both surface and cavefish populations and measured binding affinity with the recombinant human protein IRF2, as the binding domains between humans and ASCNX are highly conserved. Scientists observed that there was significant binding to the IRF2 protein at the surface fish sequence, but little to none at the cavefish sequence. This result proves that the deletion in CRE15 activates the IRF2 binding site, preventing the IRF2 repressor protein from binding to it in cavefish. After discovering that cavefish lack a repressor binding site normally present in surface fish populations, the authors tested how the presence of the IRF2 site or lack thereof affects expression of the HPDB gene. Since IRF2 acts as a repressor of HPDB, its presence results in low expression of the gene. Surface populations which possess the repressor binding site exhibit low HPDB, while surface fish with an IRF2 binding site deletion exhibit high expression, similar to the level in natural Pachon population. Pachon fish containing the restored binding site experience a dip in HPDB expression, while two intermediate mutations that convert the Pachon allele to the surface allele shift toward the expression observed in the surface allele as expected. And lastly, here's the tyrosine pathway in ASCNX mexicanus. Excess tyrosine has been reported in cavefish populations that lack melanin. Normally, melanin production demands available tyrosine, but the cavefish populations with a melanin mutation do not experience this demand. Excess tyrosine may be converted into HPDB, leading to increased expression of the gene. Further down the pathway, the excess HPDB may produce TCA cycle intermediates. The TCA cycle is the main source of energy conversion in cells, so the excess tyrosine may act as a method of energy storage and production. In conclusion, the differential regulation exhibited in CRE15 is one of several amended pathways that have led to the adaptation of cavefish populations to extreme habitats unfamiliar to surface fish. It is likely that many other differentially accessible regions identified in this study are involved in other metabolic pathways that differ between surface and cavefish. This study also provides insight into which pathways may be crucial for organisms adapting to new environments.